Hello everyone, Mr. Walker here. This lesson is going to be on the topic of enzymes, and we'll start off by talking about what these enzymes are made up of. Enzymes are proteins, at least they are usually proteins, and remember that all proteins, they're made up of the building blocks or the monomers amino acids. So when we're talking about proteins or enzymes, there can be hundreds, even thousands of these individual amino acids that are linked together. So these enzymes, what they really are catalysts, and what a catalyst does do is it increases the rate of a reaction. In the case of enzymes, they are functioning inside of or they are produced by cells, and because of that they are referred to as biological catalysts. So enzymes are biological catalysts. Overall, what they do is affect the rate of reaction, and what that usually means is that they're going to increase the rate of the reaction, and it's not just a little bit. We're talking about increasing the rate of the reaction by millions or even billions of times, and many of the reactions that do take place inside of cells, they would take place at an incredibly slow rate without having these enzymes available. Not all of the time, but quite often, and with many of the examples that we will be talking about here and come across, they will have the ending ASE. There will be some exceptions to this rule, and usually they are the older enzymes before they did use this naming format. But the example that we're taking a look at here is the enzyme lactase. So again, we're taking a look at the ending ASE that tells us that that is the enzyme. Now, enzymes are not a reactant. In the case of this reaction that we see here, our reactant that we're going to have on the left-hand side of the reactant arrow is going to be lactose. When we do talk about enzyme biochemistry, the reactant is usually referred to as the substrate. So from now on, for the most part, I'll be using the word substrate instead of reactant. So it can be one or more substrates. In this case, we have the one single substrate, the lactose. Lactose is a sugar, and again, keep in mind that quite often with sugars, we do have the ending OSE. So if you think about glucose, fructose, sucrose, lactose, like we see here, cellulose, they do have the ending OSE. Again, there are some exceptions. Other sugars that we've talked about, like glycogen and starch, of course, don't have that ending. So in this case, we have the lactose sugar, and remember that is a disaccharide. So if I just use each of these sort of circles here to represent an individual monosaccharide, in the case of the lactose, we have two monosaccharides that are joined together. So what this enzyme lactase is going to do is it's going to act on this substrate, and this is the only substrate that it can act on. Enzymes are very, very specific. You need a different enzyme for every biochemical reaction, a different enzyme for virtually every substrate. Overall, what this enzyme is going to do is grab onto the lactose and really just break this bond that is holding together the two monosaccharides. So lactose is made up of the monosaccharides glucose and galactose. So when that bond is broken, this hydrolysis reaction, aided by the enzyme lactase, is going to result in the two building blocks of lactose, which are the glucose and galactose. So you may have heard of the term lactose intolerance. If someone does have a lactose intolerance, what that means is that they are not making, or they're not making enough of this enzyme lactase. If they're not making that enzyme, that simply means that they cannot perform this biochemical reaction. They might be taking in lactose through their diet in the form of typically dairy products, and it just means that their body is then not going to be able to use it for an energy source. In order to use it for an energy source, you need to move that glucose and the galactose, the monosaccharides, from the gut and into the cells, eventually into the blood and eventually into the cells. But if you can't break that bond in the lactose, lactose is not able to move out of the gut and into the blood. So that is unavailable to those individuals for the making of ATP through cellular respiration. It does continue on further on through the gut to the large intestine, and there will be some bacteria in the large intestine. Many of these may be able to digest this lactose, and some of the products that they do produce, some of the gases that they do produce, do result in some of the discomfort and displeasure that someone who has a lactose intolerance that they would experience. So a little bit more about the enzyme structure. They're made up of amino acids, but they're not just a long chain of amino acids. What this long chain of amino acids, and again, we can be talking about hundreds or thousands of amino acids long, they do have to fold. This is really essential for the functioning of a protein and for the functioning of an enzyme. So they fold into a very specific three-dimensional structure. So if I just go back here to the initial slide, this is showing a representation of an enzyme, which again may be hundreds of amino acids in length. This is a two-dimensional picture that we're taking a look at, but in three dimensions rather, it's a very complicated structure that we do have here. So we do have to have this folding. It's not just a linear structure, a long chain of polypeptides, but it is a folded structure which does make a protein, which does make an enzyme active. So why does this folding take place? Well, each one of the amino acids, even though we didn't take a look at the properties of the individual 20 amino acids, they do differ in terms of their size. Some of them are big, some of them are small. They differ in terms of their charge. Some have a positive charge, other ones have a negative charge, and they also differ in terms of whether they like water, they're hydrophilic, or they don't like water, they are hydrophobic. So taking a look at this last example, if we do have a polypeptide chain, a long chain of amino acids, if we have a cluster of these amino acids that are hydrophilic, or if we do have groups of these amino acids at different regions of the polypeptide chain, those regions that are hydrophilic, they will tend to stick together. And not only that, they're going to be in a region where they are exposed to water because they are hydrophilic amino acids. That is different with the hydrophobic regions. These regions, again, will all kind of cluster together, but they will cluster together toward the center of the enzyme, the center of the protein, where there is no water available. If we take a look at the charge, if we do have regions that have oppositely charged amino acids, positive and negative charges, they will tend to come together. And of course, amino acids that have light charges, they will tend to repel each other. So based upon simply the properties of the individual amino acids that are making up that polypeptide chain, that is going to determine how that enzyme is going to fold. And this folding does need to be incredibly specific. So this picture at the top here, it does show just a long chain, actually not that long, but a chain of amino acids, which is called a polypeptide chain. So each of these individual amino acids that they have shaded in here, of course, are joined together by the peptide bonds. So in between each one of these where I'm drawing the lines, that is where we're going to find the peptide bonds that are joining them together. So they're showing a little bit here in terms of the different side chains that we have, and it's those side chains that are going to give the amino acids the different properties. So they do show down below there that we do have 8 different amino acids, and these amino acids, depending upon their properties, are going to determine how the folding is going to take place, going from this linear strand, this polypeptide strand, to form a much more complicated, something perhaps similar to what we see at the lower right-hand side, this folding that we see, where we have some bands, we have some spirals, and we have this very complicated three-dimensional structure. So kind of keep that in mind, because anything that does interfere with this three-dimensional structure is going to have an effect on how that enzyme functions. So what exactly is going on in terms of enzyme functioning? So when we do talk about chemical reactions, what it does involve is the bonds. It involves the breaking of bonds, and it involves the making of bonds as well. So when this happens, what we do need to do is take a look at the substrates, take a look at the reactants, and this picture that we're taking a look at here, we can see the reactants in this graph, and we can see over to the right the products. So this is a graph that you should be able to take a look at, understand what is going on with this graph. So plotted on the x-axis, what we do have is what's called the progress of reaction. So we're simply starting at the left-hand side with the reactants or the substrates, and we're ending up at the right-hand side with the products. What it's plotting on the y-axis is the amount of energy, the amount of free energy. So what this picture is showing us is that in terms of the reactants and the products, The reactants or the substrates, in fact, have much more energy than the products. So in order to go from the reactants to the products, even though the reactants do have more energy, the reactants have more energy than the products, we do need to still have an input of energy in order to reach what is called the transition state. So we do want to be able to, as it says up above with these molecules, contort and stress the bonds that might be holding together two individual building blocks in the substrates or the reactants. And in order to do that, we need to add some energy. The amount of energy that you need to add in order to reach this point and eventually lead to the formation of the products is referred to as the activation energy. So it's plotted on the graph here and it's labeled as E subscript A. That is the amount of energy that has to be added in order to eventually allow for the conversion of the reactants or the substrates to eventually form the products. So there is an input of energy, but in the end we can see that there is a difference in the amount of energy, once again, between the reactants and the products, and that is going to result in, according to this graph, a loss of energy, typically, of course, in the form of heat, a loss of energy as we go from the reactants to the products. But we did still need to have this initial input of energy in order to reach that transition state, and that is the energy of activation or the activation energy. So just a quick little point, two different kinds of reactions that we've already come across. Catabolic reactions, we're taking something big and we're breaking it down. So if we take something like a polysaccharide and break it down, that is a catabolic reaction. An anabolic reaction, really exactly the opposite. We're making something bigger, we're adding things together. Both of these may require, and typically do require in cells, the use of an enzyme. So in terms of how enzymes go about doing this, if we do have a spontaneous reaction, what that is really referring to is the fact that the products do have less energy than the reactants. So the previous example that we took a look at here, this would be considered to be a spontaneous reaction. Even though it is spontaneous, it's not going to happen without an input of energy, and once again, that energy is the activation energy. So we already know, or should already know, that if we heat up a solution, that does get the molecules moving faster. And if we're talking about a reaction that is taking place in a beaker between two or more reactants, if you do heat it up, those reactions do take place at a faster rate. However, inside of your body, inside of living organisms, there are some restrictions. So in your body, for example, our body temperature is somewhere around 35, 36, 37 degrees Celsius. That is the temperature that your body has to function at. So we can't just heat up the temperature of cells, heat up the temperature of your body in order to increase the rate of the reaction. There needs to be another method to increase the rate of the reaction, and that really is where enzymes are going to come into play. So they're going to speed up the rate of reaction without the requirement to increase the temperature. So how do enzymes go about accomplishing this? Well, if we take a look at a very similar picture to the one that we just saw, the previous one showed us the black line. So this is the course or progress of the reaction without the enzyme. So if you don't have the enzyme, in order to go from the reactants to the products, you do have to have an investment of energy, but it's a lot of energy that you have to put in. In other words, you have a large energy of activation. If you have an enzyme, this is really the key. What enzymes do is they lower the amount of energy that's required for the reaction to take place. Enzymes do not add energy to the reactants. They don't push the reactants to turn into the products. All they do is it lowers the amount of energy that is required. There is still an energy requirement, the energy of activation, but it's not as much. So these enzymes then make it much, much easier to go from the reactants or substrates to eventually forming the product or products. So how do the enzymes actually go about doing this? Well, a couple of different possibilities. First of all, they hold the reactants in a position where they're physically closer together. So these reactants in the cell might be moving about all over the place, and they may very rarely actually bump into and encounter each other. So what the enzymes are going to do is grab onto the individual substrates if there are more than one, and they're going to bring them closer together. Even if they do bump into each other within the cell, they may not bump into each other in the correct orientation. So enzymes are also going to orient their reactions. They're going to hold them in the most favorable position for the reaction to take place. And within the cell, they're going to create a favorable microenvironment for those reactions to occur. Enzymes are incredibly specific. So we already saw with the lactase enzyme that's acting on lactose, that is the only substrate that lactase is going to act on. If you have another disaccharide, like sucrose, then you need a different enzyme for that. So what reactions are going to interact with the, or what reactants rather, are going to interact with the enzyme depend upon, once again, that three-dimensional structure. The three-dimensional structure has to allow for the substrate or reactants to come into that particular three-dimensional structure. So once again, I'm sort of using these names here interchangeably, the substrate and the reactants. The active site is another big term that you do need to know. The active site is where we do have the substrate that's actually going to make contact with the enzyme. That's where the reaction is going to take place. So really important that the active site is very, very specific for that particular substrate, and that active site is maintained in terms of the three-dimensional folding. So this picture that we're taking a look at here, the blue structure that we see here is a representation of the enzyme at the left-hand side. And what it's showing is that this pocket toward the left, that is the active site. So that is the location where the substrate or the substrates are going to come in. So in this case, we do in fact have two different substrates, the red one and the green one here. So they are going to come in, they're going to bind to the active site, and when they're binding to the active site, hopefully you can see here that we do have this interaction where they're held close together and they're held in the correct orientation, and this is what is referred to as the enzyme substrate complex. So when they are brought closer together, now a reaction is going to take place, or it makes it easier for the reaction to take place because, as we talked about, it's going to lower the energy of activation and speed up that reaction. Now, after the reaction has taken place, notice that for number 4, that the products look, well, a little bit different than the reactants. They have been changed, and what that also means is they no longer really fit into the active site. So they're going to be released from the active site, and then if we follow the arrow here, this is really important with the enzymes. We've used this enzyme, we've released the product, but the enzyme was not consumed. The enzyme is not one of the substrates. It's not one of the reactants. And what that means is that enzyme, once it carries out its function and produces the products, it can once again grab onto more substrates and complete the same reaction over and over again. So one single substrate may be responsible for hundreds, thousands, tens of thousands of reactions taking place. So that previous picture that we took a look at, it kind of looked like that substrate is going to fit perfectly into the active site of the enzyme. And that's what it's showing here as well. It looks like it's going to perfectly fit in. In this case, it would, and that's referred to as the lock key mechanism. As it turns out, that's not really what happens. This is initially what they thought, is that the substrate kind of fits perfectly into that active site. In fact, what is truly the case is it's referred to as an induced fit. So this is the model that we do talk about in terms of how the substrate is going to interact with the enzyme. So if we take a look at the picture on the left-hand side, this is the active site that we see here on the enzyme. Take a look at the shape of the substrates. It doesn't exactly look like they're going to fit in here. They'll kind of notch their way into the active site, but they don't fit in perfectly. But as soon as they do come into and interact with the active site, the enzyme is going to mold itself around the substrate or the substrates. And again, that's when we're forming this enzyme substrate complex. So this molding of the enzyme around the substrate is called the induced fit model. So think of it as the substrates inducing or leading to or causing a change in the shape of the enzyme, the shape of the active site, and that is the induced fit. And then, of course, we can have the reaction taking place. Our product that we have here, in this case, we do have the two substrates that are joined together rather than split apart. That's going to be released from the enzyme, and then we're just going to go back to the beginning here again because we can use that same enzyme over and over again for multiple substrates. What are a few things that are going to affect enzyme activity? The amount of enzyme that you have, the amount of substrate that you have, or here I have the concentration of them, the temperature where the reaction is taking place, and the pH where that reaction is taking place. So if we do move the temperature away from the ideal temperature for that particular enzyme, or change the pH away from that ideal pH, what that results in is what is called denaturation. Denaturation of the enzyme. And what that really means is that that very specific three-dimensional folded structure, it's going to be lost. Denature means we have the unfolding of that enzyme. So if you unfold that enzyme, you've lost the three-dimensional structure, you've lost the active site, and it's no longer going to work anymore. When we do talk about denaturation, though, this one is a reversible change. So if, for example, we increase the temperature, that temperature is going to lead to the denaturation and the unfolding of the enzyme. The enzyme is no longer going to work because it's lost its three-dimensional structure. However, if we return it back to the original temperature, the ideal temperature, then what is going to happen is that enzyme is going to refold. So what that tells us is that the amino acid sequence contains all of the information that is necessary for the proper three-dimensional folding of that enzyme. Keeping in mind that each one of the amino acids has very, very specific properties. Those properties of the amino acids are not lost through the process of denaturation. Return it back to the original conditions, and that also leads to the refolding and the regaining of the function of that enzyme. This picture here is just showing us, again, a representation, two-dimensional representation of what could be taking place when we do have protein or enzyme denaturation. So we have, starting at the upper left-hand side here, our active site, substrate at the upper right-hand side attaching, products are being formed. At the bottom, what it's trying to show here is that if we do have denaturation, then the substrates are simply not going to fit into the active site. In fact, there may not even be an active site anymore associated with that protein, with the enzyme. Just a tiny little point here. Sometimes enzymes do require some additional assistance, and the assistance comes in the form of two different things. They are referred to as either cofactors or coenzymes. If they are cofactors, they're usually metals, like we see here, iron and zinc. And if they are coenzymes, they are typically vitamins, and these will assist in the functioning of enzymes for the proper functioning of the enzymes. A couple of graphs that we'll take a look at here. Again, very important that you're able to take a look at graphs and interpret the information in the graphs. When we see these square brackets, the square brackets just mean concentration. So substrate concentration on the left-hand side on the x-axis, enzyme concentration, the graph on the right side. So if we take a look at this, we're still taking a look at the rate of reaction, which could be measured in several different ways. It can be measured by taking a look at, for example, the use of the substrate. It can take a look at the production of the product for how fast that reaction is taking place. So what this graph on the left-hand side says, if we make our way through, moving from a low substrate concentration on the left to a high substrate concentration on the right. So initially what we see is we have more or less a straight line, a linear increase. As we increase the concentration of the substrate, that's going to increase the rate of reaction. But you'll notice that it does eventually slow down. It doesn't go down, but it does slow down, and eventually it kind of plateaus here. In this case, the reason why it's reaching a plateau, we do still have the reaction taking place, only it's kind of stabilized, and that's because in this example there's a finite amount of the enzyme. If you continue to add more and more and more and more substrate, eventually that enzyme is kind of maxed out in terms of the amount of product that it can produce, and that's where we see this plateau. So adding more and more and more substrate is not going to increase the rate of reaction anymore. You're still going to get substrate used and product formed, but the rate of reaction is going to remain consistent. The one on the right-hand side, in this case here, what we're taking a look at as the enzyme concentration. So once again, low to start off with on the left-hand side, higher on the right-hand side. Add more and more enzyme, we see that the rate of reaction is going to be increasing, and it's continuing to increase. So in this case, the only way that we're going to see this linear relationship, in other words, as the enzyme concentration increases, the rate of reaction increases. The only way this is going to happen is if you're continually adding more and more substrate as well. If this example was to run out of the substrate, then the reaction rate is going to plateau, and eventually, in fact, it's going to go down if you do run out of the substrate. But as long as you're continually replenishing that substrate, this is the shape of the curve that we would see with that example. A couple of other curves here showing things that can affect the enzyme activity or the rate of reaction. Temperature. As you change the temperature, it's going to affect the enzyme activity. Now, enzymes are very, very specific, and different enzymes work best in different conditions from different organisms. If we're taking a look at a mammal, so the curve on the left-hand side, this is from a human, and again, in a human, average body temperature is 36, 37 degrees Celsius, so that's where we're going to have the highest rate of reaction. That's where these enzymes, human enzymes, are going to work the best. So what happens if you lower the temperature? When you lower the temperature, the enzyme may not denature, but things do slow down. So you will have lower rates of reaction at lower temperatures. If you do increase the temperature, this typically will lead to denaturation, and this is why this part of the curve we see a steep drop-off. We have denaturation taking place, unfolding of the enzyme. Now keep in mind, if we bring it back to the 36, 37 degrees Celsius, that enzyme should function again as long as it is a reversible change, which is the denaturation. It also plots here that there are some other organisms that do have other ideal temperatures at what their enzymes function at. This one here, very, very hot temperature, almost 80 degrees Celsius. So this might be bacteria, well, it is bacteria that you find from a heat-tolerant environment, and that would be something like the hydrothermal vents that you find at the bottom of oceans. Bottom one here is taking a look at pH. Again, enzymes very specific. This one on the left-hand side works best at a pH of about 2. The only place that you would find a pH of 2 in your body is in your stomach, where you have the hydrochloric acid. So this enzyme, which is pepsin, it is active in your stomach. That is the only location that it is active in your stomach. It will not be active anywhere else because of the pH specificity. It only works at that pH. So if we had this enzyme, pepsin, at a pH of approximately 7, which is the normal pH of body tissues and fluids, it in fact wouldn't really work at all. The second enzyme that I'm showing you here, this one is trypsin. This is a second enzyme that's also involved in the digestive system. It is found within your intestines, the small intestine, and we can see with this one here, it also does not work best at a pH of 7, and that's because in the small intestine, it is a slightly basic environment, and that is the ideal pH for this enzyme trypsin. Both of these enzymes, by the way, they are digestive enzymes that are responsible for the digestion of proteins. A few other things that do have an impact on enzyme activity, and they're referred to as inhibitors. So an inhibitor is something that is going to lower the action, lower the functioning, lower the reactions that are taking place with a particular enzyme. So two different kinds of categories: competitive inhibitors and non-competitive inhibitors. The key to sort of keep in mind that if it is competitive, what it means is that it's competing with the substrate, actually competing with the substrate for the active site. So the Normal substrate is trying to reach the active site. The competitive inhibitor is also trying to get into the active site. So whatever you have more of, that's usually what's going to win out in terms of this competition. If there's more substrate, there might still be a sufficient number of reactions taking place, but if you have an excess number of these competitive inhibitors, which might be things like toxins and poisons, they are getting into, they're out-competing the active site, out-competing it from the normal substrate, and then we would see a dramatic drop-off in terms of the rate of reaction. Non-competitive inhibitors, a little bit different. They bind to another region of the enzyme. So we haven't talked about what this other region is. It's referred to as a regulatory site. So other than the active site, what is that region? Other than the active site, it is a regulatory site. So this regulatory site, what it does is it does change the shape of the enzyme. If something attaches to it, it changes the enzyme shape. And keep in mind, that could also involve the active site. So if that is the case, then we're not going to have, well, really any of the substrate that's interacting with the active site. So if we take a look at these three series of pictures here, the first one is normal binding, where we do have our substrate that's coming in. We have the induced fit, where the enzyme is molding around the substrate, and then we would have the reaction that's taking place. In example B here to the right, what we have is a competitive inhibitor. So we have the substrate, we have the competitive inhibitor. In this case, the inhibitor is in the active site, which means the normal substrate can't get in, and that reaction is not going to take place. It also shows on these two here that we have another region, this other pocket that we see right here. That is the regulatory site. So we can see in the last example here with the non-competitive inhibitor that it is attaching to, not the active site, but this other site. But take a look at what has happened to the active site. Even though that's not where the non-competitor inhibitor is, it has changed the shape of it. So now we're actually not really going to have any of the substrate that's now going to be able to interact with the active site. So this is a pattern that we might see with no inhibition as we increase the substrate concentration, constant amount of enzyme. We're going to have an increase in the amount of or rate of reaction, and eventually it's going to kind of plateau if we're not increasing the amount of the enzyme. If we do have competitive inhibition, well, we can still see an increase. It eventually plateaus, but it's not quite as high. And again, the more competitive inhibitors you have, the lower that plateau is going to be when it eventually reaches that stage. If we do have a non-competitive inhibitor, here it shows that we, well, in fact, do have some reaction rate. I guess that would depend upon the amount of non-competitive inhibitors, but if we do have enough of these non-competitive inhibitors that are blocking all of the enzymes, then in fact there really would be no reaction rate at all and no enzyme activity. Just a couple of other things about enzyme activity. We can also have something that's referred to as allosteric regulation of the enzymes. Here we have something that again is binding to a site other than the active site, the regulatory site, or what's referred to as an allosteric site. And what it can do is actually two different things. We can have the inhibition, like we just kind of talked about, but you can also have the opposite. You can also have an activating or a stimulating effect as well if it does stabilize the active site, making it more likely that we could have the substrate coming in. And we'll see as well in a picture here that enzymes, sometimes they're made up of more than one subunit. And if we do have this allosteric interaction, these regulators, they may in fact stabilize the subunits or destabilize the subunits, making it more or less likely that the reactions are going to take place. So in the example at the top here, we have 4 different subunits that are making up this enzyme. They're showing in this case that we have an allosteric activator. It's not binding to the active site, it's binding to the regulatory site. And when it does do that, notice that it stabilizes the active site. It maintains the active site open, so we can have these substrates that are coming in and reactions that are taking place. The examples that's at the bottom, it's really exactly the opposite. In this case, instead of an activator, what we have is an inhibitor. it still is going to bind to the regulatory site. But now take a look at what is going on with the active site. It has destabilized, it no longer looks the same, and the substrate is not going to fit into those active sites. And just another picture here taking a look at where we do have in this case an activator. We have again the subunits that are making up this multi-subunit enzyme. And here what they talk about is something referred to as cooperativity. So if we do have a substrate that binds to the active site, sorry, this one is actually not involving the regulatory site. If we have the substrate that goes into the active site, that in itself is going to stabilize the entire structure of all of the subunits, and that's going to increase the likelihood that a substrate is going to act with the other subunits as well. So the cooperativity is when you have substrate binding to one of the active sites of one of the subunits, and that's going to make it more likely that we're going to have substrate attaching to the additional active sites of the additional subunits in this larger enzyme complex. Final point here, final slide, really kind of important, feedback inhibition. Whenever there is a reaction that is taking place, your cells, your body is always kind of asking the question, was it an appropriate reaction that we did have? Was it enough of a response? Do you need more of a response? So you're always providing and always provided with feedback in terms of whatever response is made. So in this example that we see here, what we have is a biochemical pathway that's made up of 4 different enzymes, and they are numbered simply as enzyme 1, 2, 3, 4, and 5, or rather 5 different enzymes. 4 different intermediates. The 4 different intermediates are labeled A, B, C, and D. So the way this works is we have enzyme number 1, which is going to grab onto the initial substrate, which is an essential amino acid threonine. From this essential amino acid, you can make another amino acid, which is called isoleucine. That is the eventual product that we are going to have made here. But in order to go from our initial threonine to our final product isoleucine, this is a multi-step process. So again, we're going to be involving 5 different enzymes. So at the end, what we do have is our product that is formed, which is the isoleucine. Once you have enough of this isoleucine, your cells then kind of ask the question, have we made enough? Have we made too much? If there is a sufficient amount, then your cells don't really want to be making any more for a number of different reasons. You don't want to run out of the substrate, the threonine. You don't want to expend all of the energy for these reactions when you don't really need the isoleucine, and too much of something may not be good either. Too much of this isoleucine may not be a good thing. So when you do have a sufficient amount of the isoleucine, it binds to the regulatory site of, this is the initial enzyme. So when it binds to the active site, it's now going to change the shape of that active site. If it changes the shape of it, it's kind of saying we don't need to make any more isoleucine, so threonine, don't attach to the active site, and then we won't have any more of this product being made. So in essence, what we have is the final product in the chain here, the isoleucine, is going to be regulating its own production through this process referred to as feedback inhibition.